All right, guys, today let's talk about the different types of respiration. So, just to go back over the things we had done before, say when we talk about, the purpose of respiration, right? Say the purpose of respiration is to make ATP, right? Like that is our purpose for doing everything respiration. Remember, because that makes my energy for my organism, and then that energy I can use it to do cellular functions, whatever that is, like active transport or digestion or muscle movement and stuff like that. So when we talked about respiration last time, we talked about aerobic respiration. And remember, aerobic respiration, this is with oxygen. And oxygen was super duper important. Remember, he was greedy, and so he's a really good electron acceptor, right? He was able to take those electrons from the NAD and the FAD, so that way I can turn ATP synthase and make ATP, right? And so with the oxygen, this is going to help make the most ATP, right? Which really, that means this is going to be the most efficient type of respiration. Because I got to make so many, because remember from one sugar molecule, I got to make that 36 ATP. Now, the steps of aerobic respiration, right? Steps of aerobic respiration were my glycolysis, were Krebs, and then that electron transport chain. So if we recall, say, let's go back through words, say glyc meant sugar, lysis means to break, and so glycolysis, I literally broke apart my sugar, and I got to create 2 ATP from that. And then from the products of glycolysis, my pyruvates, and then the acetyl-CoAs, and then I smash those acetyl-CoAs up, I end up making another 2 ATP. But in the electron transport chain, this is where the O2 is. I got to make the most energy because that oxygen was there to take the electrons from the NADs and the FADs, and then kind of depends on how you want to count it. Let's say we got 34 ATPs, right? So again, that's why aerobic respiration is the most efficient process, because I got the most out of the sugar molecule. And then don't forget glycolysis. This was in my cytosol. And then Krebs and the electron transport chain, we won't have to name specific places, but this was definitely located in the mitochondria. That's why whenever we talk about the mitochondria is where the energy is produced. Okay? So most efficient because it has the oxygen. I can make the most ATP there. But here's the deal. When we talk about, remember the mitochondria, if I go back to it really quick, if I talk about the mitochondria, remember mitochondria is an organelle, and I find my organelle in eukaryote cells. So everything that does aerobic respiration, these are going to be my plants, my animals, and my fungi. So then here's the question, but how do prokaryotes, which are going to be my bacteria, get their energy or get their ATP? Right? Because we know bacteria are living, and if bacteria are living, that means they have to have some type of energy source. Well, how they get it is they're going to go through anaerobic respiration. Okay, anaerobic. So if we do word roots, an, that means without. And when I look at aerobic, that's talking about oxygen. So anaerobic respiration, this is a type of respiration without oxygen, okay? And so, since there's no oxygen, so there's no electron acceptor. And if there's no electron acceptor, that means I don't get to make as much ATP, right? So that means very little. Whoops, it's spelled little, right? Oh my gosh. Very little ATP, okay? And don't forget, remember, where do I make the most energy? I make the most energy in my mitochondria. And since we're talking about bacteria, they don't have a mitochondria, which means I'll only be able to do maybe 1 or 2 steps, right? So let's ask ourselves, well, what process can they do? So what process can bacteria do? They can do, let's find a nice color, they can do glycolysis, right? Because remember, glycolysis, that only happens in the cytosol. And you guys told me last time that, hey, cytosol, that's something that every living organism has, which means I can still do this process. All right, so since they have cytosol, that means they are still going to take in glucose, to take in my glucose, which means I can break it apart. But now I get to go to a new process. This new process is called fermentation. Okay, and this fermentation, because of the lack of oxygen, is going to be way less efficient. Okay, way less efficient because of this. So when we talk about anaerobic respiration, so let's write it again. Anaerobic respiration, right? I get to go through 2 stages now. Say my first stage is glycolysis, and then my second stage is fermentation. Okay, first step, second step. Now, as far as fermentation is concerned, I actually get to have 2 different kinds. I get to have alcohol fermentation, or I can have lactic acid fermentation. Kind of depends on what organism I'm looking at the moment. So if I were to write a formula for anaerobic respiration, I would still have my sugar, the C6H12O6. This is my reactant. And then I'm going to get an amount of ATP. We'll talk about that in a second. I'm still going to get some CO2 produced. And then this right here, and this part right here, where it's just an underline, this is going to depend on the type of fermentation that my organism is doing. Okay? So let's start with, actually, no, here, let's finish up this equation really quick. So glycolysis, if I look back up here, how many ATPs did I make? I got to make 2. And since I don't get to go in the mitochondria and do Krebs electron transport chain, that means in anaerobic respiration, I only get to make 2 ATP. That's why this is way less efficient than aerobic respiration. All right, let's talk about our first kind. Let's use this lovely color. So let's talk about alcohol fermentation. Alcohol fermentation. All right, alcohol fermentation. So where I'm going to see this is going to be in bacteria, and I'm also going to see this happening in yeast. And yeast is a special case because yeast is a fungi, and we know fungi have mitochondria and stuff, but yeast actually do this type of fermentation. So let's go through our first step, okay? Our first step in alcohol fermentation is going to be glycolysis, right? And remember, glycolysis, glyc is referring to sugar, which is my glucose. Lysis means to break. And so in glycolysis, I am literally breaking apart sugar. And this happens in my cytosol. And so what I'm going to do is I'm going to take my glucose. So here's my glucose. And remember, this glucose is C6H12O6. And I'm just going to concentrate on the C's because, you know, organic chem. So 1, 2, 3, 4, 5, 6. And then what do I do if I want to get energy out of this sucker? Is I'm going to break it apart. And when I break it apart, I get 2 3-carbon molecules. So let's start with what's important first. When I break something apart, I get energy. This is where I make my 2 ATP. And I still get to make my 2 NADHs, right? But this right here, this is what I want at the moment because I don't have an electron acceptor, so I can't break that NADH apart to make more ATP. All right, so I broke him apart. Since he's broken apart, that means he has a new name. Don't forget, both of these molecules right here, these are called my pyruvates or pyruvic acid. Say they both the same name. Then, after that, because there is no oxygen available, I go immediately into step 2. And step 2 is fermentation. And so what I'm going to do in fermentation, which is still, guess what, happening in my cytosol, what I'm going to do is I'm going to take those 2 pyruvates, right? I'm going to take those 2 pyruvates and I'm going to turn them into something else. And since this is alcohol fermentation, I'm actually going to turn those into alcohol, right? In alcohol, this is where we get beer. This is where we get bread, right? Say those types of things, because remember, bread comes from yeast. And so since I don't get to move on and go on to Krebs and electron transport chain, I only get to earn, let's find a nice color, that means I only get to make 2 ATP. And if I'm only making 2 ATP, this is way less efficient because my other process, aerobic respiration, I get to make 36 or 38, depending on how you want to count it. This one, I only get to make 2. So very, very inefficient process. But that's really all alcohol fermentation is. I take my sugar. Once I take my sugar right here, I break it apart. I get those 2 ATP. Then I take these, turn them into alcohol, and then I'll just have to do the whole process over again. That's why it's very inefficient, because I have to have a lot of sugar and I don't get a lot of bang for my buck. All right, let's talk about our next process then. Next process is called lactic acid fermentation. In my lactic acid fermentation, this is going to happen in human muscle cells, okay? So let's start the process as we always do. Let's start with glycolysis. Now, we already know glycolysis means to break sugar. Don't forget that glycolysis is happening in my cytosol. So I'm going to go through the same process. I'm going to take my glucose, which is the C6H12O6. So I'm going to write it out like this, 1, 2, 3, 4, 5, 6. To get the energy out of this sucker, I am going to break him apart. When I break him apart, that is going to give me 2 3-carbon molecules. What's important about this is that when I break him, I get 2 ATPs. That's what I want because that is the energy source. I'm also going to get 2 NADHs, but I don't get to do a lot with him because I don't have oxygen to accept his electrons anymore. And then as a result, I get to make these 2 guys. Don't forget, these are my pyruvates. So now I get to go on to my next step, which is fermentation. Don't forget, fermentation's still happening inside my cytosol. And so my human muscle cells, what they're going to do is going to take these pyruvates, and then they are going to turn this into lactic acid, right? Again, because I only earn 2 ATP because of glycolysis, this is way less efficient. This is why all of our cells don't do lactic acid fermentation, because we wouldn't have enough to live. But it makes sense, because lactic acid, that's the stuff that builds up inside your cells when you exercise. Because if we did a nice little flow chart, like let's say you guys are exercising. Well, when you guys exercise, that means you're going to use up, or you're going to lose a bunch of oxygen, right? Because you're breathing really hard. And so then when you start to breathe really hard and you run out of oxygen, say you run out of oxygen, that means your cells your muscle cells, they're going to switch to fermentation, or anaerobic fermentation. And then when you guys switch to fermentation, right, that's when you start to make that lactic acid inside your cells. And if you can imagine acid inside your cells, doesn't feel real great. And so when you switch that lactic acid, that's when your muscles just start to burn, right? Like they start to hurt so bad. Well, guess what? When they start to hurt so bad, what do? That means you start to slow down. Because if you slow down, guess what? You get to catch your breath. And then when you catch your breath, that means you get to regain the oxygen. And when you guys regain your oxygen, then you get to go back to aerobic. Go back to aerobic respiration. And that's when the burning stops, right? So we all know about lactic acid. Maybe we didn't know what the name of it was. But take home message from all of this. So if we go back through everything, aerobic respiration is my most efficient process because it has oxygen, and that's when I get to make the most ATP. Anaerobic respiration, this is going to be without oxygen, right? And since it's without oxygen, this is going to be way less efficient. And these are going to be my bacteria and my yeast cells. I have 2 different kinds of anaerobic respiration. I have alcohol, I have lactic acid. In alcohol fermentation, the bacteria and the yeast, they break apart that sugar through glycolysis, and then they switch to fermentation where alcohol is a product. And in the end, I only get to make 2 ATP, which is super less efficient. And then in muscle cells, I get to do the same process. I get to do glycolysis. And then when I run out of oxygen, I switch to fermentation, which makes my cells make lactic acid. I only get 2 ATP, which is super less efficient. So in the end, let's go ahead and make a nice little chart super quickly. When I talk about respiration, there are 2 kinds, okay? I have aerobic, and then I have anaerobic, right? Aerobic respiration, this is with oxygen. Anaerobic, this is without oxygen. When I have oxygen, that means I get to take place in the cytosol and the mitochondria. Whereas anaerobic respiration, this is only going to happen in the cytosol. Aerobic respiration, this is the most efficient, and I get to make a lot of ATP, so we'll say the 38 ATP. Anaerobic respiration, less efficient, I only get to make 2 ATP. In aerobic respiration, I get to go through 3 steps. Glycolysis, Krebs, electron transport chain. In anaerobic respiration, I only get to go through glycolysis. That's why it is way less efficient. And then in anaerobic respiration, these are going to be all eukaryotes, so plants, animals, fungi, versus anaerobic. These are going to be prokaryotes, which are all bacteria, and then yeast cells and human muscle cells. Okay, there you go. Say, I think if you guys know this, you're going to be pretty well set. Say, hope that helps.